So this is chemical digestion. This is the chemistry of the physiology of digestion. So digestion is a catabolic thing. It's taking complex food molecules and breaking them down into their simpler substances that can be absorbed. It requires the use of enzymes to lower the energy of activation and drive those chemical reactions. And all of those chemical reactions are hydrolysis. It's basically adding a water molecule into the bonds that cause the bonds to break. So you add an H to one side and an OH to the other, and you cause the chemical breakdown of the food molecules, the complex food molecules. So I've brought this up in class. I've talked about it in the other sections, but we'll cover it in detail here. So carbohydrates are a very important food molecule. And so that may be starches, it could be sugars, but somehow they are these rings of monosaccharides joined together, and we have to break the rings up. So we use things called amylases. So salivary amylase starts to break down the complex starches. It breaks the bonds so that the starches get broken down into smaller, more manageable units. If you leave it with saliva long enough, it will actually start to become disaccharides. It'll start to become sugars, sucrose, maltose, and that kind of thing. Most of the time, though, you just break down the starches into smaller units of starch. In the duodenum, it mixes with pancreatic amylase, and pancreatic amylase's job is to break the starches down into disaccharides. So the polysaccharides go down into disaccharides, and basically there's dextrin, there's sucrose, there's maltose, there's lactose. Are the main ones. So really, they are some sort of combination between the monosaccharides, the glucoses and the fructoses. And it's the monosaccharides that we need to absorb. So on the brush border of the entire small intestine, there are enzymes that are part of the cell membranes that break down those disaccharides into the monosaccharides to facilitate them being absorbed. So each enzyme is responsible for a different disaccharide. So sucrose is broken down into glucose and fructose by sucrase. Maltose is broken down by maltase. Lactose is broken down by lactase. If a person doesn't make lactase, they can't break down lactose, and so that lactose will not be absorbed. It will end up going into the large intestine, where there are plenty of bacteria more than happy to break down that lactose for you and turn it into gas. They'll happily eat it. So these brush border enzymes are responsible for that final breakdown to the monosaccharides. They get absorbed into the bloodstream, right? And they get absorbed, co-transported with sodium via secondary active transport. There's some facilitated diffusion, and then these are all carried to the liver, where they are dealt with. Some of them are allowed to stay in the bloodstream. Others get turned into glycogen and stored, etc., etc. So the brush border enzymes break lactose, maltose, and sucrose into galactose, glucose, and fructose um, by whatever means that, they need. I don't really care about the paths of absorption with each of the different ones. Um, when we break down proteins, the initial protein breakdown happens in the stomach with pepsin. It changes these large proteins into smaller polypeptides. Then the proteases, the trypsin and the chymotrypsin and etc., break down these polypeptides into very small peptide bonds, right? And then the brush border enzymes finish breaking them down into the amino acids. So if the dipeptases break down one peptide bond, etc., and then the amino acid is secondary active transport, so it's coupled to sodium. Okay, so that's what gets absorbed. So maybe some dipeptides, but they're small molecules. Lipids get broken down, but before they can get broken down, because they're not water soluble, they tend to form globs in the chyme. And so we emulsify them to create more surface area for the enzymes to work on. Now, there's a little bit of lipase that's secreted in the mouth, in the saliva, salivary lipase, that starts to work when it gets down into the stomach in that acidic environment. And it starts to break down the lipids there. But most lipids wait until they are expelled into the duodenum from the pylorus and get mixed with the bile, and then the bile emulsifies it, and then enzymes from the pancreas, pancreatic lipases, start to break them down into glycerol and fatty acids that we can use. Now, they don't need a brush border enzyme because they're not being broken down that much. They get absorbed into the villi, and if they end up in capillary blood, they end up going to the liver via the hepatic portal vein. If they combine with carrier things, with lecithin, and they end up entering the lymphatics via these special lymph capillaries called lacteals, and then they're brought to systemic circulation up at the right lymphatic duct and the thoracic duct. They enter into the brachiocephalic veins and then go to the heart and end up in circulation that way. So we're looking at unemulsified fats get emulsified, then they get broken down and then absorbed. Nucleic acids are hugely important. So the pancreas secretes ribonuclease that breaks down RNA, and deoxyribonuclease, which breaks down DNA. And there's RNA and DNA in everything that we eat because it's cellular in nature. It's absorbed via active transport, and then it's brought to the liver and processed there via the hepatic portal vein. Are you seeing a pattern here? Everything ends up going to the liver via the hepatic portal vein. We break down these long chromatin particles or strands into smaller by these ribonuclease and deoxyribonuclease. And then there's a brush border enzyme that breaks it down into the A's and C's and the T's and G's and U's to get absorbed and stored and then used by mitosis everywhere in our cells. Now, the other foods that we bring in, the other nutrients that we bring in, are vitamins. There are fat-soluble vitamins that are carried with the fat, and then so they're absorbed at the same time as the fat. The water-soluble vitamins are absorbed passively or with transporters. B12 needs this intrinsic factor to bind with to make it into. So it's unique. But this is one of the reasons why, like if you look at vitamin A, vitamin D, vitamin E, to a lesser extent vitamin K, but we get vitamin A and D, or vitamin A and E especially from vegetables, green leafy vegetables. This is why we want to eat them with fat. This is why you put butter on carrots. This is why you put salad dressing on salad, because it facilitates the absorption of these fat-soluble vitamins that are found in these plants. The water-soluble vitamins are C and B, and they absorb pretty easily because they are water-soluble. So like I said before, the flora in your large intestine manufacture vitamin K and some B vitamins. So that's how they pay their rent for a nice place to live. That absorption happens in the large intestine because that's where these vitamins are really manufactured. Electrolytes get absorbed anywhere in the small intestine. So iron and calcium are absorbed in the duodenum. In order to absorb calcium, you need vitamin D. Vitamin D is synthesized in the skin from cholesterol because of sunlight, and it facilitates the absorption of calcium. Sodium is kind of everywhere. Potassium, etc. 95% of the water, and there's a lot of water because there's water that we ingest with the food. There's water that we drink. There's water that is secreted via the various juices that the stomach secretions, the pancreatic secretions, the intestinal secretions, all add water to the chyme, and it all has to be reabsorbed so we don't lose it. There's water created by hydrolysis or used by hydrolysis. So there is kind of some metabolic water as well. So it's absorbed by osmosis. Good rule of thumb in physiology is if there's ever a question on how does water move, the answer is probably osmosis. Most of it happens in the small intestine. The large intestine reabsorbs the last 5%. And then the malabsorption syndromes are about reasons why you can't absorb. So like for instance, gluten damages the intestinal villi, damages the brush border, so then you don't get that final brush border enzymatic action, and those nutrients can't be absorbed. But that's all part of pathology and not covered in this course.